Hello, my name is Nikhil Lawrence, and today I will be discussing cell adhesion. So, to start off, cell adhesion is very important when it comes to assembling cells into tissues. However, cells simply do not just stick together just to form tissues. They need adhesion, some type of adhesion in a distinctive pattern in order for them to form tissues. So I'm going to start off with talking about two different types of tissues in the animal cells. These two different types of tissues are epithelial tissues and connective tissues. Epithelial tissues consist of two ranges of the cell. The first one is the apical range. This is at the top of the cell. And this region comes in contact with the outside environments, which obviously you can see because it is at the top of the cell. Now moving on to the basolateral region, right under this tight junction that I'll be going more in depth later on in the presentation. So the basolateral region is basically the surface that meets the basement membrane, which is actually part of the basolatinum. So moving on to the corrective tissue. Compared to the epithelial tissue, the connective tissue is less loosely organized. And by being loosely organized, it has their cells. Their cells are actually just embedded into the extracellular fibers, as you can see right here, how they are in pretty much embedded everywhere in the extra as epithelial tissue. They're pretty organized and together. So moving on to cell-to-cell junction. So in order to form tissues, cell-to-cell junction is needed. So pretty much cell-to-cell junction is the function of joining or pretty much attaching cells together. And there are various types of cell-to-cell junction. First, I'm going to start off with tight junction. As you can see here, tight junction is part of the apical region. This acts as a watertight seal between adjacent cells. As you can see here that the cells are tightly bonded together. And the reason for this bonding is so that it keeps any fluid or ions or molecules from getting through these cells. And this helps to actually protect the cell. So moving on to adherent junction. The adherent junction is basically one type of adhesive junction. It's pretty much attaching to pretty much a way of basically it bonds tightly to other neighboring cells and attaches to the actin, basically the F actin. I'm sorry, excuse me. Next, moving on to the gap junction. The gap junction acts as similar to like a pathway or a type of tunnel between two cells. And this lets fluid and other ions and molecules flow in and out using a chemical and electrical communication. In some cases, it may be the spread of action potential. Now moving on to desmosomes. Desmosomes act as a small disc or a spot wheel. It adheres two cells together, and this way is able to connect to the intermediate filaments such as keratin. And lastly, we have excuse me if I pronounce this wrong, humidisome. It adheres the bisal surface to the, basically seen here, it adheres the bisal surface to the bisal lamina, and it actually attaches to the intermediate filaments. Now, this is just a table showing the types of cell-to-cell and cell matrix attachments. So, shown for the cell-to-cell adhesions, I pretty much talked about them in my previous slides, such as gap junction, tight junctions, adherent junction, and so on. However, one that I really didn't focus on yet is focal adhesion. We can see focal adhesion is the cell to extracellular matrix adhesion, and its structure is associated with the actin microfilaments. And I'm going to talk more in depth about focal adhesions later on in my PowerPoint. So this just gives a really tiny brief overview of what I'll be discussing later on. So moving on to cadherins. Cadherins are calcium-dependent cell adhesion molecules that are located within adherent junctions. So looking at part A of the figure, you see that a cadherin, which in this case a E cadherin was used, has a extracellular domain, which is right here, that extends outside of the cell to interlock with a neighboring cell, which can be shown here. And the extracellular domain is in a belt or a zip-like structure. You can also see that the beta cation binds to the tail of the cadherin, where you see that the alpha catenine binds to the beta catenine, And from here, both of these proteins are able to link the cadherin to the cytoskeleton. And also we have the P120 catenine, which is an adherent junction that binds to the tail of the cadherin, just like the alpha catenine, but it is able to mediate cadherin's stability and row activity. Shown in part B below is just a amino stain of a MDCK cell, which you can see in the green is pretty much a structure of a cadherin. In this case, in the green is the E cadherin. It's just showing the buckle-like structure that it has on the extracellular domain when it interacts or when it interlocks with a neighboring cell. So now moving on to the effect of adherin on cell adhesion. So here, actually an experiment was done to test the effect of cadherins on cell adhesion. Culture L cells, which are fibroblast cells, were used because they do not normally attach to one another. So E adherent, and P adherent were added to these L cells. And when these cadherins were added, these E and P cadherins started to actually start being produced in these cells, which can be seen here. They were producing these cells, and you can see that those cells that began producing, which is producing pre-cadherin, which is in the red, they began to bind to other cells that produce p-cadherins. Same vice versa with the E cadherins. You see that whatever cells produce E-cadherins, they bind it to cells that produce E-cadherin, and you see that they are all basically attaching to one another. Now moving on to cadherins during embryonic development. Here is a picture of two frog embryos. Cadherins plays an essential role in embryonic development. In frog embryos, when messenger RNAs are depleted, EP cadherins proteins aren't produced anymore. They just stop being produced, and this actually results in the loss of normal organization of the embryo. So as you see here, see here in part A, excuse me, you see that the control embryo doesn't have any, actually has the EP cadherin protein, so it has a normal structure. As you see here, the structure is distorted and abnormal, and this is due to the EP cadherin, the EP adherent deficiency. So moving on is the desmosomes. So as previously stated before in my earlier slides, desmosomes acts as a spot weld to join two cells together and attach to keratin filaments. I'm sorry, excuse me. And desmosomes are typically found in tissues that has constant stress and pressure, which is typically is in the skin. Say that. So desmosomes typically do rely on cadherins, and the types of cadherins that are found in desmosomes are desmocollins and desmoglins. So shown in part A of this figure, you see a electron micrograph of a desmosome which joins two cells together. Part B shows a better imagery of what part A is trying to show. So you see at the top, there are two neighboring cells at the plasma membrane. You see that the linker protein binds this cytosolic tail, then links them to the cytoskeleton. The plankoglobin, which is a beta-catenin protein, part of the beta-catenin protein family, binds to the desmocollin cadherin. The plankoglobin binds to the desmoplankin protein, and the plankophyllin, which is part of the P120 catenin protein family, binds to the cadherin in the desoplankton, and this is where, and it can also stabilize desmosomes. So moving on to the types of extracellular matrix. So the extracellular matrix is a collection of molecules that hold the cells of tissues together. And there are different types of extracellular matrix. First is the bone. The bone is a hard and rigid extracellular matrix with very, very many scattered cells. Next, we have the cartilage. The cartilage is way, way more flexible than the bone extracellular matrix, and it contains a lot of protoglycogen. And lastly, the connective tissue, which is usually underneath the skin, and it has a gelatin-like extracellular matrix that contains scattered fibroblast cells. Now moving on to the structure of integrins. Fibronectins and laminins allow cells to attach to the extracellular matrix. Fibronectins serve as a bridge that joins cells to the extracellular matrix. Um, they can bind to animal cells by specific receptors, such as a transmembrane protein, which is called which is an integrin. Basically, an integrin are composed of two large transmembrane glycoproteins consisting of the alpha and the beta subunit. That forms a non-covalent heterodimer. The alpha and the beta subunits extracellular portion, which is shown up here, it interacts to form a binding site for a specific protein. As however, the alpha and beta cytosolic portion, which is actually found right here, the integrin has a binding site for linker proteins that can actually join the cytoskeleton to the extracellular matrix. So in the figure shown, this is actually integrin is a fibronectin receptor. So the integrin has a fibronectin binding site at the outer membrane, which is shown here by the alpha beta subunits. And at the cytoplasmic membrane, at the alpha beta subunits, it has a linker protein binding site, which is binding site for taline. Then it can be shown here that the alpha subunit splits due to a disulfide bond. So lastly, integrins in cytoskeleton. Integrins do not directly adhere to the extracellular matrix, but they link to the extracellular matrix by focal adhesion and hemidysosome. Shown in the part A of this figure, focal adhesions consist of a cluster of integrins that forms a linkage between the F-actin bundles to the extracellular matrix. Linker proteins such as talent, which can be shown here, can bind to the vinculin, which is a actin binding protein, and the alpha actinine that joins the integrin to the actin cytoskeleton. Shown here in part B is basically an invasive human breast cancer cell that was strained for actin microfilaments, which can be shown right here in the red. And the blue shows the vacuoline to indicate integrin attachment sites. And as you see, which is shown in the blue, and as you can see that the F-actin actually ends or terminates at the focal adhesion, which you can kind of see at the ends. Moving on to part C, which is the hemidysosome. These are epithelial cells that attach to the lamina in the basal laminae. Integrins attach to the intermediate filaments, such as the keratin. The linker proteins form plates that connects the clustered integrin to the cytoskeleton. In the Parkins attaches the keratin filaments to the integrin. And then the transmembrane proteins BPAG2 and BPAG1, which is the plankin, actually serve as a bridge right here between the keratin and the laminin. And D just simply shows an extracellular surface of a heminidesosome. It just shows that the heminidesosome is pretty much right next to the biasolamina. So this concludes my presentation on cell adhesion. I hope you learned something, and I hope you enjoyed this presentation, and thank you for listening.